From a culture tube, an environmental sample, down to a contaminated food, bacteria are present. They are invisible to the naked eye, yet they influence every environment on Earth. What happens when you need to know how many individual bacterial cells are thriving in a certain environment? You need some method for counting the bacteria accurately. But a liquid culture of bacteria may contain a billion cells in every milliliter of media. Think about it. If you count one bacteria in every second, it would take you decades to get to 1 billion. Obviously, it is one tedious job. We only want to count between 30 to 300. So, how do we get there? The answer is serial dilution. A serial dilution is a series of sequential dilutions used to reduce a dense culture of cells to a more usable concentration. Each dilution will reduce the concentration of bacteria by a specific amount. So, by calculating the total dilution over the entire series, it is possible to calculate how many bacteria you started with. Before starting the serial dilution, a flask or beaker which contains the stock solution must be readied, as well as, in this example, 5 test tubes which each contains 9 ml of diluent. Each test tube must be labeled accordingly. From the flask, get 1 ml of the stock solution and mix with the diluent in test tube 1, acquiring a total volume of 10 ml. From the test tube 1, acquire 1 ml of the aliquot and mix with the diluent in test tube 2. And from test tube 2, get again 1 ml of aliquot and mix with the diluent in test tube 3. This procedure is repeated for the test tubes 3, 4, and 5. From the dilution method, we are able to calculate the dilution and dilution factor in every step. The formula in solving for the dilution and dilution factor are as follows. In our example, we have 1 ml of aliquot and 9 ml of diluent. For test tube 1, we get 1 over 10 or 10 raised to negative 1 dilution and a dilution factor of 10. For the test tube 2, we get 1 over 100 or 10 raised to negative 2 dilution and a dilution factor of 100. The calculation for the remaining test tubes goes on. After the dilution, counting the number of colony-forming units or CFUs comes next. So, how do we count the CFUs? We do this by obtaining 1 ml of the solutions from every test tube and putting it in the agar plates. For a 24-hour cycle, the CFUs appear and the counting is done. However, in the earlier dilutions, counting may not be possible. Hence, it is very important to dilute the stock solution further. After counting the number of CFUs, we can then calculate the bacterial density of the solution given with this formula. Here is an example problem. The dilution is done in 3 trials. To calculate the bacterial density in every step, get the average of the number of CFUs in every step. Substitute the known variables and then obtain the result. To conclude the final answer, the largest bacterial density is the correct one. However, in this example problem, the second largest density is chosen because the number of CFUs in the last dilution is less than 30. Remember that when the number of CFUs is less than 30, the result may be subjected to errors, especially when the solution becomes contaminated.